Everyone, and welcome to your Chapter 20 video. If you are watching this in the fall of 2020, a few announcements, and even if you're not watching this in the fall of 2020, I am sure that whatever semester you're watching this, you are drawing to the end of your course because Chapter 20 is the last chapter or two we will talk about. Making sure I have everything. Okay, so what lies ahead of you? For fall of 2020, folks, here's what you've got left, okay? So you're going to have a homework that I'm going to post this week. However, I am extending the due date. This homework will be a little bit different, and it'll be over a gene editing technology called CRISPR. Okay, you may have heard of this. All right, so homework will be due of a little bit later. Check Blackboard for that. Again, fall of 2020 people, you also have a quiz, and that will be due at the end of next week, which let me double check the date on that for my fall of 2020 peeps. That is going to be November 20th. Okay. And then at this point, sometime this week or next week, I will be posting your final exam study guide. You still have some time before the final exam. Your final exam will be December 4th, fall of 2020 people. But I'm going to go ahead and post your study guide so that you can get a jump on preparing for that. All right. Let's move on and talk about Chapter 20, regardless of the semester that you are watching this. And I think that this is a chapter most people are going to really enjoy. Up until now, I've given you a lot of information that's been very theoretical. And many of you may be thinking, this is all well and good, but you probably have a career goal in mind, and you might be struggling to see how some of this information relates to your career aspirations. The nice thing about Chapter 20 is it takes the theoretical information that we have gained over the last few chapters and shows you how we apply it today in a field called biotechnology. So how we use information about DNA replication, genetics, all that transcription, translation, all that good stuff. We're going to apply it now in this biotechnology chapter. And in this chapter, we are going to cover really 4 topics. I'm going to break this into probably 3 lectures, okay? And over the course of these 3 lectures, we're going to cover 4 different topics. Today, we're going to talk about something called gene cloning, and I'll add to that not just gene cloning, but recombinant DNA technology. The next lecture, which will be posted in the next day or 2, is going to be over the polymerase chain reaction and how we analyze that, which is a process called gel electrophoresis. And then our last lecture will be over a really hot topic right now, which is on gene editing using a system called CRISPR. Usually it's CRISPR-Cas9. You may have heard of it. And I will also just mention briefly some information on analyzing genomes, okay? So let's focus in and zero in on the topic of the day, which is this topic of gene cloning or recombinant DNA technology. So I think the best way for us to address this topic is for us to tell a little bit of a story about the medicine insulin, okay? So before I start talking to you about insulin, let me define for you this term recombinant DNA technology, okay? If we are recombining DNA, what we're doing is we are combining DNA from 2 different organisms. Okay, so we're going to be taking DNA from humans, and in this case, what I'm about to describe to you today, we're going to insert some human DNA into bacteria. That's going to make the bacteria something called a transgenic organism. And a transgenic organism is an organism that has DNA, oops, hold on, has DNA from 2 or more species. So we're going to take, in this story that I'm about to tell you about insulin, we're going to take the human insulin gene, the human DNA, and we're going to insert it into E. coli, thereby making E. coli transgenic. All right. So this idea is we're going to be recombining DNA, and we're going to do that through a process called cloning, which I'll define for you in just a minute. Okay, so first, let's talk a little bit about the hormone insulin. You probably have heard of this before. Insulin is a hormone that is produced in the pancreas of many, many organisms. We find it all throughout vertebrates. And what this hormone does is it regulates the glucose in your blood. So if you eat a big meal, for example, I had a wonderful meal yesterday, macaroni and cheese, vegan cheese. You guys know I don't eat dairy. Macaroni and quote unquote cheese, and I had green beans and corn, and it was just delightful. And when I ate that meal, as a result, there was a lot of carbohydrates in that meal. My blood sugar went up. And so what insulin does is it binds to the receptor on the surface of some cells. In particular, you find these receptors in the liver, in your fat tissue, in your muscle tissue. When the insulin binds to these receptors on the surface of a cell, you can see here it's bound, it opens up glucose channels, allowing glucose to be taken up into the cell. And once it's taken up into the cell, it can be processed for storage, so it can be turned into things like glycogen, or it can be converted into fat. So it can be processed and used, or it could also be converted into ATP via cellular respiration. But the thing is, glucose is no good to you if it's in your bloodstream. It's got to get inside your cells in order to be useful. Now, if you have a problem where you lack insulin, then you have a disease that you've probably heard of called diabetes. So what happens in diabetes, there's 2 possible things. You could lack insulin and not make enough of it. That's called type 1 diabetes. You may have also heard of type 2 diabetes. And type 2 diabetes is where your receptors don't work so well. They've become insulin resistant because you eat too much glucose. Usually type 2 diabetes is addressed with diet and exercise. If you lose weight and sort of get your health under control, you can often bring your type 2 diabetes under control. Type 1 diabetics simply don't make enough insulin. And so in order for them to be able to bring this glucose into their cells so that it's useful, they actually have to supplement with extra glucose. They have to take extra glucose. They have to supplement with extra insulin, and they have to take extra insulin. So sorry, I messed that up. So you have to take extra insulin if you're a type 1 diabetic so that you can get glucose into your cells. If you don't do that, the glucose stays in your bloodstream and eventually makes its way out through your urine. So you may be familiar with people who have to have their urine tested for sugar to determine whether or not they are managing their diabetes. That's actually how pregnant women get checked for something called gestational diabetes. We look for glucose in their urine because if it's in the urine, it's not getting taken up into the cells. Okay, so that's how insulin works. Now, what if you're someone who does not make sufficient insulin? Then you need to take supplementary insulin. Where is this insulin going to come from? Insulin is a hormone. It is a peptide hormone, meaning that it is a protein. And we know that proteins are encoded in your DNA, right? So that must mean that there is a gene that tells your body how to make the protein insulin. So for a long time, the source for insulin for diabetics was we would process the pancreas of pigs or of cows to get pig or cow insulin, and people would inject that. And it was good enough. It was good enough. It was similar enough to our insulin that it was effective. But everyone agrees that it would be ideal if, one, we didn't have to worry about processing these pig and beef pancreas because that involves, possible contamination and things like that. Also, it would be ideal if humans could take human glucose. So with the advent of recombinant DNA technology, what we can do is we can take the gene for human glucose, or sorry, I keep saying glucose, the gene for human insulin, and what we can do is we can insert this gene into bacteria. The bacteria can then transcribe and translate that gene, and we can trick, so to speak, these bacteria into making human insulin. So that's how insulin is made today. We don't really use the pig or the beef insulin so much anymore because it's really, really easy for us to culture bacteria and then separate out the human insulin and purify this human insulin, okay? So today what we're going to cover is how we go about inserting this human gene into a bacteria. To understand this, we're going to need to review a little bit about bacterial genes. So if I draw you a bacterium, and I'm going to draw this sort of as a rod-shaped bacterium, remember that they have a single circular chromosome, okay? But you might remember from early studies in this course that sometimes bacteria have additional genetic material. They're little bitty circular pieces of double-stranded DNA that are called plasmids, okay? So these plasmids exist in the bacteria in addition to the main chromosome. And the plasmids are interesting. They don't really contain genes that are necessary for growth, reproduction, or basic cellular function. These plasmids contain sort of extra genes, things that are, not necessary for the cell to survive, but it's nice if the cell has them. One great example of a gene that you find on the plasmid of bacteria is one for antibiotic resistance. So antibiotic resistance isn't necessary for the normal functioning of a bacterium, but it sure is a nice thing if you're a bacteria to have this gene because it makes you able to live in environments where there might be antibiotics. So in order for us to make this human insulin, we are going to use recombinant DNA technology to perform gene cloning. And here's a very simplified version of what gene cloning is and what it does. The most basic definition of gene cloning is that you're producing many identical copies of a gene. That's where this term clone comes from, right? If something is a clone, it's usually an identical copy. So in gene cloning, we're going to make many identical copies of a gene. And in this case, we're going to be making identical copies of the human gene for insulin. And the way that we're going to do that is we're going to take our target gene, which is the human insulin gene, and we're going to take a bacterial plasmid, which is what I've got for you right here. We are going to use a cut and paste mechanism where we cut open the plasmid. So you can see we're going to cut it here, and that's what you see here. This plasmid has been cut. We're going to paste in the gene of interest. So here's our insulin gene. And then we're going to actually weld this together, okay? So now what we have is what's called a recombinant plasmid that has both the original bacterial bit, which is what I'm outlining for you here, and the human insulin bit, which is what I'm drawing for you here, okay? This cut and paste mechanism, we're going to use two different molecules to perform this cut and paste. The cut is going to be performed using something called a restriction enzyme. I'm going to tell you how these restriction enzymes work. Your book calls them restriction endonucleases. That's fine as well. And then the pasting, where we actually are able to join this gene here to the original strand, we've actually met this molecule before. This pasting is done by DNA ligase. You might remember when we studied DNA replication that DNA ligase is capable of welding together the sugar phosphate backbone, and I'm going to put BB for backbone, on a molecule of DNA. Where we saw it used most often was to join Okazaki fragments. Well, it turns out it can join a lot of different types of fragments, including we can use that enzyme to join fragments in the laboratory. So we can join these fragments here to make this recombinant plasmid. Then all that's left for us to do is to insert this plasmid into a bacteria, okay? So let's talk about how we might do this. Let's first focus in on how restriction enzymes work. So restriction enzymes were first discovered in bacteria, and we believe that so let's talk about how we're going to get that gene into the plasmid using those restriction enzymes that I mentioned. So we're going to talk exactly about how these restriction enzymes work. Let's take a little closer look at a plasmid. So on this plasmid, I want to point out some features. First of all, as I mentioned, this plasmid has an antibiotic, I'm just going to put AB resistance gene, and we're going to use that a little later, okay? So on this particular plasmid, we have an antibiotic resistance gene, and we have this sequence that I want to draw your attention to, okay? So let's talk a little bit about these restriction enzymes and where we think they came from. We first discovered restriction enzymes in bacteria, okay? And what we think that they were originally intended for was to act as a defense mechanism against foreign genetic material. Y'all will remember that we've talked before about how viruses can infect bacteria. And viruses, the entire way that they work is by injecting their genetic material into a target cell and then tricking that cell's machinery into transcribing and translating their genes, turning them into proteins and making them into new viruses. So basically it just overtakes the machinery of the host cell to make more copies of itself. So you can imagine that restriction enzymes, which their whole deal is that they cut DNA, right? A restriction enzyme would be a very useful tool if you were a bacteria trying to defend yourself against foreign DNA from viruses. That's where we think these restriction enzymes originated. So, but today we use them in the lab as sort of the cut part of a cut and paste mechanism. And there are many, many different restriction enzymes. The one that I'm going to tell you about today is perhaps the most famous of them. It's called EcoR1. Eco stands for E. coli. R stands for restriction enzyme, and 1 stands for the first one we characterize. Now, even though there are many, many different restriction enzymes, they all work in the same way. So let's talk about that. What these restriction enzymes do, as I alluded to earlier, is they come along and they patrol the target DNA until they reach a specific target sequence called the cut sequence, okay? So I'm just going to write cut-seq. In this case, the cut sequence, as I mentioned before, for this particular restriction enzyme is GAATTC. Sorry, I have not mentioned that. Okay, so the cut sequence for this particular enzyme is GAATTC. Now let's write out the complementary sequence for this sequence. C, T, T, A, G. So what's going to happen when this restriction enzyme comes along and recognizes this cut sequence is it's going to make a cut. EcoR1 cuts between the G and the A, and it does that on both strands. So what you end up with is sort of this lopsided cut. Let me erase here, which is indicated for you in your book diagram by this dashed line. Now let's envision what would happen if we pulled apart these cuts on this circle, okay? If we pulled the cuts in this direction and in this direction, okay? So let's pull these apart. This first segment here, what would it look like? It would be a G and a C that are paired. And then you would have an unpaired region, TTAA, okay? You would have a gap here that was unpaired, all right? Now, let's pull this cut in this direction. What you would find if you looked at this segment and you pulled it apart is you would have an A, T, T, C with a G down here. But all of this region, if we pulled these cuts apart, would be unpaired. That means that it is open for complementary base pairing. And we actually call these sticky ends. And that is, believe it or not, the formal name that we use in molecular biology to describe these open cut sites. So let's look at it on this diagram here. Let me get rid of this. Okay. So here, what we see is that our EcoR1 has made its cuts, and these cuts have been pulled apart on this plasmid, leaving open a region here and here available to complementary base pairing. Now I'm going to go back to this slide for just a second. Now, let's also say that in the lab, what we've been able to do is isolate the gene sequence, the mRNA sequence, actually. We're going to say that this is the sequence that's already had the intron spliced out. So this is our sequence for the human insulin protein, okay? Even though it's the mRNA sequence, we have converted it to DNA, and we call that cDNA. And the reason that we convert it to DNA is because we want to insert this into another DNA molecule, and we need the molecules to match. So we've taken the mRNA sequence and we've converted it to DNA. But just know that this is the same thing as the coding strand that we've mentioned before, and why it was so important to understand those differences. So this is that DNA that looks just like the mRNA, okay? And we've made it double-stranded, and we're going to put it on in, okay? The other thing that we've done after we've isolated this human insulin gene in the lab, we have added on either end the sequence that is recognized as the cut site for EcoR1. What that means is that if EcoR1 makes its cuts on this linear molecule, let me erase this so we can see. On this linear molecule, EcoR1 is making cuts. Now, let's pull off the cuts. And I'm going to do that by actually just blacking this out. And I'll black this out. You can see that, again, we're going to have some sticky ends. We're going to have a region here that is unpaired, and we're going to have a region here that is unpaired. Let's look at it on this slide. Here's an unpaired region, and here's an unpaired region. Now, what I'm hoping you realize is that we cut both of these, both the plasmid and the target DNA, were cut using EcoR1. They were cut using the exact same restriction enzyme. I hope you can see that the sticky end on this end of our target human insulin gene complements the sticky end here, and that the sticky end on this end complements this sticky end here. So let's look here. Can you see that these line up beautifully? Now that you have complementary base pairing, now we've performed our cut in our cut and paste mechanism. Now all that we need to do is to zip up the sugar phosphate backbone, and that the pasting mechanism happens using DNA ligase. Okay, let's look at how this looks like in an animation. So I'm going to pause this recording, and when we come back, I'll be looking at an animation from your textbook. Okay, so this is the textbook this is the textbook page that talks about recombinant DNA technology, and here is an excellent animation that describes what I've just shown you.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So once we have our human insulin gene inserted into this plasmid, all that's left for us to do is to take these little individual plasmids that we've made and engineered and stick them into a bacteria, okay? Once these plasmids get inside these bacteria, here's the really neat thing, and here's where this antibiotic resistance gene that's on this plasmid is going to be important. So you may have noticed that these plasmids were all labeled with an antibiotic resistance gene. So we have this end up here that we've engineered, and we have this end down here with an antibiotic resistance gene. If we can slip these plasmids into some bacteria, and then we can grow them in a petri dish or in some bacterial broth that contains antibiotics, then only the bacteria that have the plasmid inserted will live, right? Because theoretically, what would happen is any bacteria that didn't get a copy of this plasmid would be killed by the antibiotics. So as these bacteria grow and divide, the only bacteria that will grow and divide are those that contain our gene of interest, our insulin. So all of these E. coli cells are making our genetic clones. They are making exact copies of this plasmid right here that contains our human insulin gene of interest. Furthermore, these bacteria are transcribing and translating that gene, and so they are making our insulin that we can then use a whole other suite of biotechnology tools to isolate from the bacteria and package as medicine. Now, you might have a couple of questions about this process. For example, you might be wondering how we get these plasmids for use in the laboratory. or how we might isolate them if we wanted to. Like, let's just say that you had gone through this whole process and you'd grown up a bunch of these E. coli and you were interested in getting your gene or your plasmid back out. Well, we can actually separate plasmids from bacterial DNA using a process of lysis. Lysis followed by centrifugation. Now I'm going to oversimplify this process. I'm just going to put centrifuge. Okay. Essentially, when you're looking at a bacterium, and I know this is not the best resolution, but this was the best, most simple diagram that I could find. So in your bacteria, what you have is the cell wall and cell membrane. You have some proteins, you have some DNA, which is this black bit here, and you have the plasmids. What we can do is we can use a very strong base that will both disrupt the cell membrane. It'll crack open the cell membrane, and it will denature the proteins. It denatures the proteins and the big DNA chromosome. I'm going to put big chromosome. So basically, this base, remember that bases have strong negative charges. These negative charges are going to mess up the cell membrane, creating holes in it, if you will. They're also going to mess up the binding of DNA and the binding of proteins. Now, what's going to happen is that later on, we're going to restore the proper pH. And our little bitty plasmids are going to be able to recombine, but the big stuff stays denatured. So what we can do is we can put this guy into a centrifuge, and basically a pellet of all the big stuff will form at the bottom. So this is the big chromosome as well as the protein that was in the bacterial cell. Also, any of the cell membrane stuff. So basically all of the cell junk goes down to the bottom of a centrifuge tube if you spin this really fast in a centrifuge, and you end up with a pellet, right? But these plasmids are really small. And because they're really small, they stay dissolved in the part up here, which is liquid, which is what's called the supernatant. So what you can do to get these plasmids essentially is you just treat these bacteria with a strong base. You then basically centrifuge down what you've got. The pellet at the bottom is all of the trash that you don't need, but the liquid at the top is going to have your plasmid, and you just suction this off and you have the material that you need to do this whole process, which is pretty cool. Now, once we have this plasmid and we've made it like we want it to, I said that the whole key is that we have to insert these plasmids into the bacteria to get them to grow. And you might be wondering how we go about inserting the plasmids into some bacteria. So that is a process called bacterial transformation. And basically all that we do to make that happen is I'm just going to put poke holes into the bacterial cell membrane. We can do this using strong chemicals, or we can do this using electricity. There's something called an electroporator. But essentially you treat these with a chemical and then you freeze these cells up. And when you do that, even though you've poked holes in this cell membrane, when you freeze these guys down, the cell membranes contract, the holes seal up, and everything's fine. If you then thaw these cells out and you heat them up, and you put them in a solution that contains your plasmids, the holes are big enough that the plasmids can get in. You then give these bacteria some time to repair their cell membranes under ideal growth conditions. So that's the recovery phase. That'll seal up those holes. And once you've done that, you've transformed your bacteria by adding some DNA that you engineered in the lab. You can then plate these guys on a petri dish, and you can plate them on some agar, which is what this stuff here is called. It's called agar. You can plate them on some agar that contains antibiotics, antibiotic positive. That means that any of these bacteria that did not take up a plasmid would die. Only those that took up the plasmid will live. So each of these little bacterial colonies represents a bunch of cloned DNA. Now, what can we use this DNA for? So once we've gone through this process, we can then do some things with these plasmid. Oh, here we go. Here we go. We can do something with these little bacteria that have their DNA and then their engineered plasmids inserted in here. Now, this whole story, I've used this example of using this system to make insulin. So we have inserted the human gene for insulin into this bacterium, and in so doing, this gene can now be transcribed and it can be translated to make the insulin protein. And we can go through different protocols to purify that insulin, and we can use that insulin as a therapeutic for people with type 1 diabetes. So this is now a situation where we can give human insulin to human patients. We can also use this system to make a bunch of copies of a gene of interest, because every time these bacteria divide, they're making exact copies of themselves, and when they divide, their plasmids are dividing as well. So we can use this as a way to clone or make exact copies of our gene of interest. Another thing we can do is use a process very similar to this to create genetically modified organisms. And in particular, today, I want to talk to you a little bit about genetically modified organism plants, okay? So you've probably seen in the supermarket, if you go to Food Lion or Harris Teeter or something like that, you've probably seen things that are labeled as GMO free or no GMO, something like that. And so you may have gotten the idea that genetically modifying organisms somehow poses a health hazard to you if you purchase a GMO tomato, for example. But that is not quite the case. So let's talk a little bit about genetically modifying plants. In the process of genetically modifying plants or animals, what we're doing is we're directly altering the genes of livestock and crops. So here you can see we have our bacteria and a plasmid, right? And we have a gene of interest. This gene of interest might be a gene that is an insect resistance gene. It might be a weed resistance gene, a weed, or we'll say a weed killer resistance gene. Okay, we'll put resistance. So these are genes that might be beneficial for a plant to have. So for example, if you had a gene that you knew would cause a plant to not be eaten by insects, you might be interested in inserting that gene into a crop like, say, corn. If you had a gene that you knew made corn resistant to the effects of weed killers, then that would be awesome because you could insert that gene into corn. And then if you sprayed a cornfield with weed killer, you would not affect your crop, but you would be able to keep the weeds down and increase the space that your crop has for growing. So a very similar process is used where you find a gene of interest, you genetically engineer a recombinant DNA plasmid, you insert that plasmid into a bacteria via the process of bacterial transformation. Then you grow up these bacteria. And the key is that you use a bacteria that you know can infect plants. So you allow these plants to become infected. You actually allowed their seeds to become infected with these various genetically modified bacteria. And thereby you can transfer your gene of interest to your crop. You can transfer it to your corn or whatever it is that you're looking at. So some examples of genes that we insert into corn or other plants. RRC stands for Roundup Ready Crops. Roundup Ready crops are crops that are herbicide resistant. Herbicide resistant. And what does that mean? Well, that means that if you spray them with a weed killer, which is what an herbicide is, the crop itself will not die, but all of the weeds around it will die. Another example of a gene that we use is called the BT gene. That stands for Bacillus thuringiensis. Now, that is the name of a bacteria from which we have derived a gene, and this gene forms something called, it encodes something called cryproteins. Oops, these cryproteins make the plant very, very distasteful to insects. You can kind of think of it as making the plant sort of like a ghost pepper for insects. So insects stop eating these plants. All right, so these are examples of genes that we've actually used and inserted into corn, sunflowers, soybeans, other products of interest. And I have on here, because I used this in a different class before, to know the vocabulary transgenic organism, which is just an organism that contains genes from other species. And a transgene is the gene from another species that's being inserted. Now let's look at some of the different genetically modified foods that we have actually devised. So for example, here's one called golden rice, and you can see that this rice is actually, come on there. I don't know why it's doing this. Yeah, there we go. You can see that this rice is actually golden in color, and that is because it has been genetically modified to produce the vitamin beta carotene. So beta carotene is what you find in carrots to give them their orange color. If you add a gene to make beta carotene in this rice, you have boosted the nutritional value of this rice. You know that rice is a staple product, a staple crop in many societies. And while it is very calorie dense, it's got lots of carbohydrates and lots of calories, it doesn't always have a ton of vitamins. So this particular genetic modification could help sort of assuage or alleviate some of the nutritional problems that we see globally. Another crop that we have is something called BT cotton. Remember, BT is that gene that makes things insect resistant. So most of the cotton that we grow around here is BT cotton, okay? There is a virus resistant papaya, Roundup Ready alfalfa. Let me show you a couple more. We have BT corn, and you also have Roundup Ready corn that you can plant. We have biotech soybeans, and the list goes on and on. It turns out that something like 90% of corn and soy that's grown in this country is genetically modified, okay? And something like 70% of your processed foods that you eat, things like, Doritos and so on, 70% of your processed foods contain genetically modified organisms. Now, as you probably know from looking at the packaging, say if you go shopping at the grocery store, there's all this, anti-GMO packaging. These are GMO free, and if they're GMO free, they cost more. And so that implies to a lot of folks that GMOs must pose a health risk. But the truth is that it's a much more complicated story than that. Genetically modified organisms have both pros and cons associated with their use. So let's talk about a few of those. Some of the benefits to having a genetically modified organism, often they are drought tolerant. Okay, they have been genetically modified to tolerate periods of dryness. They might have increased nutrition, like we saw with that golden rice. We might increase production of our crops by introducing genes that make them, insect resistant or something like that. And we also lower production costs. We lower labor costs. So ultimately, all of this is a good thing because it contributes to something called food security, whereby we can grow more food than we could without the benefit of these genetic modifications. So in thinking about like what some of the benefits are, studies have been done, and we found that it turns out the benefits of planting, say, for example, genetically modified corn, where maybe you have insect resistance and you have herbicide resistance, turns out that if you plant these varieties, you increase your crop yield something on the order of 22%. So you get 22% more corn out of your crop if you're planting these genetically modified varieties. You also increase your profits by something like 68%. Now, why would that be? Well, you've increased your yield, but you've lowered your production costs. For example, if you are planting corn that is Roundup ready, that means you don't have to hire someone to weed your cornfield. You can instead just spray an herbicide like Roundup to get rid of the weeds. It also means we see a decrease of 37% in chemical use, and specifically in insecticide use. So if you have inserted a gene that makes these plants insect resistant, you don't have to spray a whole bunch of harsh chemicals on these plants to inhibit insects from eating them. And that's actually really good for the environment. It's good for human health as well. It also means that we have a decreased need for new farmland. So there's lots and lots of benefits for new farmland. Now, I don't want to paint too rosy a picture. There are also costs associated with the use of genetically modified organisms. Now, I told you on the earlier slide that if we're talking about insecticides, you decrease the use of insecticides by 37%, right? But one of the problems that we see with genetically modified organisms is that it actually increases the use of herbicides, of weed killers. Because if you're planting a crop that is weed killer resistant, then you can actually just be very liberal and spray a whole bunch of weed killer in your fields. Your corn will be just fine. You'll get rid of the weeds and you'll increase your yield. We actually see a 39% increase in herbicide use if you plant a genetically modified organism. Now, why is this important? Well, if you had a rain event, some of that herbicide could run off of your agricultural field, run into adjacent regions, and kill some of the native plants that are there. There's also some worry about health outcomes that are associated with GMO consumption, like if you're eating genetically modified organisms, we have no evidence at all that there is any sort of human health concern with eating a genetically modified organism. One other concern is that there could be ecological risks associated with planting genetically modified organisms. Now, what might those ecological risks be? Well, it turns out that plants are very good at hybridizing with other species. So let's say that you had some corn that you planted that was Roundup ready. It was herbicide resistant, so Roundup ready corn. And let's say that some pollen from that corn actually fused with the egg of some weed. So we'll put some pollen here. And it fertilized a weed. Well, here's the problem. If this Roundup Ready gene, if this weed killer resistance gene ends up getting transferred to a weed, you could create a super weed that could take over an agriculture field and not be controlled using herbicides. Now, while this is theoretically possible, we have never seen evidence of this in the wild. Okay, we have no documentation that this has ever happened with a genetic modified organism. In addition to these concerns, there's been a huge public debate over the extent to which we should be using genetically modified organisms. One concern is that our global food supply is basically being controlled by the companies that generate these seeds for the genetically modified corn, soybeans, etc. So some common companies you might have heard of are Monsanto, Syngenta, and there are others. And the idea is that these are the people who are producing these genetically modified organisms. They sell the seeds for these crops to farmers. And the idea that these large corporations are kind of in control of our global food supply does make some folks quite nervous, okay? Another issue that arises is this idea of what's called pollen drift. And what happens in pollen drift is if you have two fields that are adjacent to one another, one being an organic field with no GMOs, so I'll put organic, no GMOs. And then this one over here that is planting genetically modified corn or something like that, the pollen from this field could easily blow into this field. If that should happen, and this farmer should seek some kind of a certification that he isn't growing, non-GMO corn, he might be denied that certification and unable to market his product for the higher prices that these types of products usually demand. So I tell you all of this as a practical application of some of the stuff that we talked about in this lecture. And I also tell you this because I feel like there's a great deal of misperception about some of these products. So as you decide whether or not to spend the extra money on a tomato that is non-GMO, you can sort of make some decisions as to whether or not you think it's worth the extra money, or are you willing to eat the tomato that has been genetically engineered, but might be a little less expensive. All right, next time we talk, we'll be talking about the use of the polymerase chain reaction in biotechnology. And until then, I'm here if you have any questions, let me know. Talk to you soon. Bye-bye.